Good afternoon. I'd like to welcome you to today's Medical Center Hour. I'm Marcia Day Childress from the Center for Biomedical Ethics and Humanities. We're pleased to see all of you here today. This is a program called Experiment 11, Dark Secrets Behind the Discovery of a Wonder Drug. We live now in an era when antibiotics against dread and deadly diseases are ubiquitous, all but taken for granted, and when drug discovery is a powerful global economic engine. But consider how it was just 75 or so years ago, when penicillin was new and not yet widely available, and when the search was on for similar, even more effective and more broadly efficacious antimicrobials. In 1943, Albert Schatz, a young doctoral student at New Jersey's Rutgers Agricultural College, was working on a wartime project testing bacteria from farmyard soil when he discovered streptomycin, a new antibiotic that was the first effective agent against tuberculosis, which had claimed 2 billion human lives in the previous 2 centuries. As it happens, the story of streptomycin involves much more than just the discovery of a life-saving drug. It turns out it's a story of scandalous behavior by Albert Schatz's senior professor, Selman Waksman. It's a cautionary tale about claiming or getting credit, rightful or not, and reaping benefit or not for scientific research. This saga has continuing repercussions for research ethics, for academic science, for university enrichment, for patent law, and for the evolution in the US and across the world of big pharma. Seasoned journalist Peter Pringle wrote this book, Experiment 11, to probe this gripping story of professional misconduct and so much more. We're happy to welcome Mr. Pringle to this Medical Center Hour, which is one of our History of the Health Sciences lectures, to explore with us Experiment 11 and the lessons of the dark secrets behind the discovery of a wonder drug. Mr. Pringle lives and writes in New York, having worked earlier in many different parts of the world as a journalist. You'll find a brief bio sketch in your handout. I should mention, too, that Mr. Pringle has no financial conflicts of interest to disclose. So we're most grateful for partnership on Mr. Pringle's visit and this talk with the Health Sciences Library's History of the Health Sciences Lecture Series. And now, Peter Pringle and Experiment 11.
Well, good afternoon. I'm honored to be here today at the Medical Center Hour, and my invitation came last summer from Joan Klein, and I'm very sorry that I never had a chance to thank her in person, but I do thank Marcia and also Dan, I guess he's here, from the Health Sciences Library for making it also easy for me to come here. I'm having a lovely time, despite the rain. That's one of the covers, but you must be wondering slightly. I'm not wearing a white coat. I don't have a stethoscope around my. And so, what on earth am I doing here on such a particular subject about which a journalist should know very little? So I'm just going to have a little fun to start with, just to introduce me, if I can operate this thing. Yeah. Okay, so this is the missing link that you're all looking for between me and this book. It's an ammonite. We were discussing last night with some students about ammonites. I see they're here today. I'm pleased to see them. I was a paleontologist for about 6 months after I left university and realized that this is somewhat of a dead end and that it would probably be better for me to go into journalism. If you look at this beautiful creature, you see these striations, they're called, on the sides here. This is an evolutionary process, and they change through the years. So my experiment in those days was to get a handle on the Jurassic Sea. This is 175 million years ago. We'll come up to the present. And to see if that sea covered the south of Britain, Normandy, and went all the way over to the Caspian. And it's sort of up there. So that is the Jurassic Sea as is extended. There's Africa and all that. Not looking quite the same. And anyway, so we went out there and we hacked at these rocks and got these ammonites out and looked at these striations, and we found out that yes, indeed, it was true. So suddenly I became a discoverer, and you know, it's a great elation to be something like that. But this is a much more serious story, which I'm now going to tell you. In order to explain my guiding light in all my work, actually, well, let's leave the Jurassic. Margaret Mead, everybody knows who she is, once had throughout her life a very stern maxim which said that the only thing worth doing in life is to add to the sum of accurate information in the world. So I looked up to that, and I've tried to do it. And this business with streptomycin was trying to find what was really accurate between what one side was saying, which was that the professor had discovered streptomycin, and what the other side was saying, which was that the student had discovered streptomycin. Okay, so here we go. The students in the two protagonists are here in chummier times, I have to say. This is Schatz and this is Waksman at Rutgers University. Of course, disputed discoveries, another little diversion perhaps, are well known in science. And perhaps we should just have a quick look at Pythagoras. Who knows that he came up with this theorem? I mean, we accept that he did. But was it the Babylonians or the Chinese or the Indians perhaps, maybe? I mean, had they figured it out centuries before? How can we be so sure? Isaac Newton and Gottfried Leibniz. Did Isaac Newton actually figure out calculus? Well, a lot of people say there's a very good case for Gottfried Leibniz. Next, as you guessed ahead of time, was it Darwin who's keeping a secret in the corner there? Or was it Alfred Russel Wallace, who came up with the theory of evolution? Who invented the radio? Marconi, Tesla, or was it Alexander Popov? Actually, I jumped for Alexander Popov. He's a terrific Russian person. And of course, you know, generally speaking, there is Stigler's law of eponymy. I'm sure you're all very familiar with that. Basically says that no scientific discovery is named after its original discoverer. The really nice thing about Stigler's law, he was a Chicago statistician, by the way, was that he was not the person who came up with this law, but it was named after him. The actual person who came up with this law was Robert Merton, also of Chicago. He of such notable concepts as unintended consequences and self-fulfilling prophecy. But in scientific discovery, one tends to forget that researchers who unearth great facts about nature are like the rest of us. They're only human. So they indulge in all kinds of human failures, greed, dishonesty, vanity, etc. And one of the greatest books that I think has ever been written about the scientific work and scientific investigation is by an Australian, of course, called Ian Beveridge. who was an animal pathologist, who became the director of the Institute of Animal Pathology in Cambridge in England. He wrote a marvelous book in the 1950s called The Art of Scientific Investigation. And the unforgettable quote that I'm going to show you now from him, there you are. Complete honesty is, of course, imperative in scientific work. So now let's go on to the dishonest part. I don't know, I mean, today we talk about scientific ethics and we talk about, are they worse than they were 10 years ago? Are they better? How should we learn about this? Is it just a bunch of lawyers telling us, creating for us, encouraging us to find loopholes in it, or what is it? And we're often told that this misbehavior, which I see it as actually, it is misbehavior, results from systemic problems of the job, such as the pressures to publish. Or another excuse in the internet age is that journal papers are scrutinized by more readers and inevitably more mistakes, deliberate or otherwise, are found. So let's go on to talk about streptomycin, but in streptomycin case we are not talking about mistakes. We are talking about deliberate distortion by a professor of the work of his student. And this was done under the rules of the academic etiquette of the day, rules which said that the lab director always took the laurels. In the history of science, claims of rank were not only common but accepted, especially in Europe. The professor was a godlike figure. Some of them still are. As a result, myths that were created about credit, who had actually done what, sometimes credit was wrongly applied by nothing more than droit de seigneur. The professor's name always came first on the journal paper publishing experiments, and no student had the audacity to challenge the professor. Researchers were lucky to be mentioned in the footnote of the paper. So here is the dramatic and I think quite moving story of streptomycin. It started at the beginning of the Second World War. The hunt was on for new antibiotics that cure the diseases of war, the massive bacterial infections in battle wounds, and the epidemics that arise in crowded and devastated cities from diseases such as typhoid, cholera, and tuberculosis. The first antibiotic, which you all know, and here he is, was discovered by Alexander Fleming in 1928. Nothing much happened until 1942 when they really needed penicillin, and so they started producing it. But penicillin will only cure some bacterial infections, as you know. No antibiotic had yet been discovered from the other tougher bacteria, the ones that cause typhoid and cholera, the deadly diseases like tularemia, and the most deadly killer of them all, TB. Antibiotics are chemical compounds excreted by bacteria and fungi. Fleming discovered penicillin by accident when spores of the fungus Penicillium landed in a petri dish in which he was growing Staphylococcus. Before World War II, scientists in Russia and Europe had noted that one of the most prolific producers appeared to be a group of bacteria called the actinomycetes. These are, sorry I don't have a picture of them, but they're thread-like things. They're sort of halfway on the evolutionary scale between a bacterium and a fungus. And Selman Waksman became, by dint of his origins, an expert in these actinomycetes. And they're found in the soil if you ever walk through a wood after rain, right, there's a sort of woody smell that's produced by these creatures. And here is Selman Waksman. So he was born in Russia, fled from Tsarist Russia in 1910, took his degree at Rutgers, PhD at Berkeley on enzymes, and by World War II, He had risen in the Rutgers ranks to become the head of the new department of microbiology. Most of his research was on the effect of bacteria on the soil, because Rutgers was an agricultural college, how they break down the minerals to form humus. But the war brought new priorities. Waksman's small band of researchers soon started finding a handful of antibiotics from microbes in the farmyard soil right outside his laboratory. The process of discovery was simple enough. Silly simple, one of the students called it. Fill a petri dish with moist soil, drop some known harmful bacteria into the soil, and wait for the telltale clear zones to appear where the antibiotics are working to kill the bacteria. Under the right conditions, these bacteria produced antibiotics that killed the germs that cause infections, but they also destroyed human cells and were too toxic for general use. So while this thing was going on, we're in 1942, huge pressure for the production of penicillin and for the discovery of new antibiotics. And along comes Albert Schatz and his family. He was born in 1920, so he's 22 years old. He's done his first degree, he's doing a PhD under Waksman. And his parents also come from Russia. And it's curious that the actinomycins actually were first really looked at in any meaningful way by the Russians. And of course, Waksman's great advantage was, and Schatz's too, because he grew up speaking and learning Russian, was that they could read the papers that were the scientific papers that were telling, that were the only scientific papers at that time on actinomycins. So he joined Waksman's staff at Rutgers, but his service was cut short when he, and then he got drafted, of course, and he went to the Army Air Corps. And during that time, he worked in a hospital in Miami, and he saw all these casualties coming home from the front with diseases for which there was no cure, simply no cure. And so he determined that if he ever got out of the army, he would go back and start to see if he couldn't find antibiotics which could help. He goes back. There's actually everybody's having a nice time with Waksman. They called him their great white father. He was very nice to them. Albert Schatz is living on a small stipend of $40 a month in a makeshift room off the campus greenhouse, and he's working day and night. He has a kind of bee in his bonnet, you might say, about finding something. And with remarkable good luck, within 3 months, he found a candidate microbe. It seemed to kill bacteria penicillin would not touch. But what Schatz could not know was, without further experiments, was whether the antibiotic would also be toxic to animals and to humans. So they had to have more tests. In January 1944, Schatz wrote up his initial results for a science journal. Waksman added his own expertise to the article and generously put Schatz's name first, an acknowledgement that the moment of discovery of streptomycin actually belonged to Schatz. Then Schatz started to test streptomycin against the human TB microbe, an experiment so risky of infection that Waksman would not go near the laboratory where Schatz was doing the tests. And he discovered that it worked. Oh, that's him in the army, sorry. Here are his test tubes. If you go from left to right, it's full of microbacterium, and then it goes, and the last one is almost clear, except for so he had found it. But of course, they were in no position in Rutgers to do any animal experiments. So they called in the Mayo Clinic, and two great guys called Feldman and Hinshaw did most of the testing on guinea pigs at the Mayo Clinic. And by the end of the summer of 1944, so a year after streptomycin had actually been discovered, they confirmed that it was in fact really effective against TB. So, Waksman and Schatz signed a patent application, each swearing on oath that they were co-discoverers of streptomycin. At this point, they're getting on quite well. Schatz is doing all the grunt work, and Waksman's sort of organizing where streptomycin will be produced and where it will be tested. And the patent, according to Waksman and what he told Schatz at that time, would be owned by Merck. The reason for this is that Merck has its headquarters just down the road from Rutgers University in Rahway. And when Waksman didn't have any money, Rutgers didn't have any money to continue the research, Merck put up some money. So on the condition that if they discovered anything, it belonged to Merck. So the patent, the first patent for streptomycin belonged to Merck. And everything seemed to be fine. Waksman said, we're not going to make any money out of this, told Schatz, we're not going to make any money out of this, this is for the war effort, this is what's going to happen. But then things just changed rather dramatically. Waksman was about 57 at the time. He was basically at the end of his career, towards the end of his career, and although he established himself as a leader in the chosen field of microbiology and an expert in the actinomycetes, he had not achieved the fame and fortune that he really sought. The papers in the archives at Rutgers and the Library of Congress, which I've seen, show that he became absolutely determined, obsessed is probably the right word, to become known as the sole discoverer of streptomycin, so that he could be considered for a Nobel Prize. After all, I mean, Alexander Fleming with Howard Florey and Ernst Chain had received the prize, the 1945 prize for medicine for the discovery of penicillin. Why not one for streptomycin? Waksman also sought a share of Merck's royalties, and nobody knew anything about that at this time. So in order to help himself along this path that he had chosen, he began to exclude his student, Albert Schatz, from the discovery. And when reporters came to Rutgers to interview him and to find out about streptomycin, how it was discovered and where and when, Schatz was edged out and kept in his basement lab. And we know this was deliberate because at some point he told one of the other PhD students who asked, why are you not including Schatz in this process? He said, because Albert is far too immature to take the fame and the fortune I have to take it. At the same time, Waksman began to tell a version of the discovery that questions part of Schatz's work. In his basement lab, Schatz had actually isolated 2 strains of actinomycetes that produced the antibiotic. But Waksman now suggested that it was he, Waksman, who had somehow been directly involved in the isolation of the 2nd strain. But that was not true. And documents in the Rutgers archive make it clear that Waksman wanted Schatz simply to go away, or to be certainly out of the way. And so he arranged, without Schatz's knowledge, for the patent to be transferred from Merck to Rutgers. The reason they had to do this was because Merck George Merck, who was then the president of Merck, had a conflict of interest. He was supposed to be producing penicillin and antibiotics, but he was also secretly the chairman and the organizer of the US very early biological warfare committee. This was top secret work, I'm not allowed to talk about it, but the conflict was that, well, look, you know, if you're the only, if Merck is going to be the only producer of streptomycin, then this is going to be a limited quantity that you can produce. So you have to let this patent go, and you have to let all the pharmaceutical companies that there were, not very many of them, have a go at it and produce as much as you can. So Rutgers set up, with Waksman's knowledge, set up a special foundation, I guess you have on here, patent foundation, to administer the patent, and put Waksman in charge and agreed to pay him 20% of the royalties, again, without telling the student Schatz. But as Schatz was technically co-discoverer, Waksman had to get Schatz to sign over his patent rights originally given to Merck to the university. So one day, according to Schatz, Waksman called him into his office and asked him to sign the papers transferring the patent to Rutgers. Waksman told Schatz that neither of them would profit from the transfer. The money and the royalties would go directly to Rutgers. That was simply not true. The royalties soon started flowing. Waksman got his 20%, and within a very short period of time, he was earning tens of thousands of dollars when his salary was only $10,000. As he got richer, however, he did have some pangs of guilt. So Schatz at this point was over in California doing some research and living on his GI stipend, GI Bill stipend, $100 a month, with a wife and babies on the way. And so Waksman started sending him checks for $500, a lot of money, you know, 5 times the amount that he was earning. And he sent him 3 checks for $500. And each time the student Schatz wrote him a note saying, you don't need to do this, I'm okay. You know, be lovely to have some more money, but you don't need to do this. And on the 3rd check, which they just kept coming, he wrote an anguished letter to Waksman saying, something's going on, I don't know what it is, but you know, it's obviously not right. So could you please tell me what's going on and don't send me any more checks? And instead of writing back a letter saying actually what's going on is the following, I'm earning a bundle on this patent and I want to share it with you, and this is how we can do it, and why don't you come back to Rutgers and spend a weekend together and we can discuss it and, you know, do the decent thing. Instead of doing that, he foresaw something that was not going to be any good for him, Waksman. He saw that he couldn't bring himself to tell Schatz that he had lied to him. He couldn't bring himself to tell Schatz that there was an arrangement that he had which was making him money. So instead of doing that, instead of having a reasonable conversation, he accused Schatz of grandstanding, of making out that his contribution to the lab research was more than it ever was, and accusing him literally in a letter of merely being a pair of hands at the lab bench, nothing more. And he had no right to any more recognition than he'd already been given for the discovery, and that anything that Rutgers had done or was doing in terms of the patent was none of Schatz's concern. Well, oh, and the little threat at the end of that letter, horrible letter, was that if Schatz continued to complain about any part of this, then the next letter he would receive would be from the Rutgers lawyers. So Schatz was in a bit of a panic, but he had, you know, a threat of legal action, and he had a rather wily uncle who was, you know, one of those dentists who weren't really dentists. You know, they practiced in the back rooms of pubs and stuff like that. Anyway, street smart, you might say. And he said, well, why don't you sue Waksman? I says, sue my professor? How could I possibly do that? I don't have any money, and it could ruin my career because the clergy will disapprove me to such an extent, the scientific clergy, I mean, to such an extent that I'll never get a job again. And so the wily uncle said, no, we can fix the money part of it, because in New Jersey we have these trial lawyers. And trial lawyers will take on your case, and you've got a really good case, and you will win, and they will take 50%, 60%, 70%, but you'll win, you'll become co-discoverer of streptomycin, you'll get some of the action from the patent, and everything will be rosy. Well, that's exactly what happened. And they won the case, and Schatz was awarded something like $12,000 a year, which was a much lower percentage, obviously, than Waksman. And Waksman had also included all the researchers who had not discovered streptomycin to have the same amount as Schatz. So he just wanted to keep this thing on a certain level, not shining the light on Schatz at all. And the reason for that, we should now go into. Because Waksman had one more thing that he needed to do, and that was to get a Nobel Prize. So he started, he was a terrific self-promoter, so he started going around Europe. He started Waksman Institutes in various countries, funded by the patent money, and dropping in every now and again to the Karolinska Institute in order to see how he was doing. And he got nominated several years in a row, but nothing actually happened because the Nobel Prize committee really wanted, they'd heard about the student, Schatz, and they really wanted to know what was the exact relationship between Schatz and the professor. So, and there are letters back and forth, strange to relate actually now, that the Nobel Committee actually wrote to Schatz to ask him about that relationship, rather than look at the lawsuit, which of course had, you know, chapter and verse and lots of evidence and all the rest of it, as how Waksman had actually behaved. So eventually the Nobel Committee, they believed Waksman's version of it, and in the process, once again, Waksman was saying, you don't need to pay any attention to Schatz's performance here, it was just a grunt bench worker. And in 1952, Waksman is awarded the Nobel Prize for medicine and physiology. Actually, for the discovery of streptomycin, that was the original citation. And Schatz then complained, and so did some of Schatz's friends, and he was working at a small national agricultural college in New Jersey at the time. The president wrote a letter to the king, blah, 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 all that. But they wrote back and said, I'm sorry, you know, once a Nobel Prize has been awarded, it cannot be taken away. So you can't just go back and say, oh, hang on, there's 3 other people who need to be included here. But what they did do, which is quite interesting, they actually said, the citation changed from the original one announcing the award to the one actually at the presentation, because you know there's 3 months period between October and December when it happens. And so the one actually produced at the ceremony was ingenious, systematic, and successful studies that have led to the discovery of streptomycin. So Nobel Committee getting themselves off the hook at the same time. And Waksman, unrelenting, he had a 6,113 word acceptance lecture that did not mention Albert Schatz and listed him only in an appendix, number 12 out of number 19 of other researchers who had worked under Waksman, half of whom had not been at Rutgers when Schatz discovered streptomycin. So, and Waksman's legal team weighed in very heavily at that point and said that Schatz had behaved, quote, like a dog yapping at the heels of a great world figure. Of course, this was not the first time the Nobel Committee had come under fire for its selection of a winner, so I'll just have a little diversion here to remind you of these events. This is Scientific American's top 10 Nobel snubs. Dear old Dmitri, who never got it for the periodic table. Charles Best, you all know, never got it for insulin. Lisa Meitner, never got it for nuclear fission. Rosalind Franklin, never got it for the structure of DNA. Jocelyn Bell Burnell, never got it for the discovery of pulsars. And the 10th person on this list was Albert Schatz. So you would think that that would kind of be the end of the story, and it should be in a minute. But there's one more thing. After the Nobel Prize, Waksman went from strength to strength, of course, winning more accolades, and his position in history was assured. The Smithsonian Institution in Washington, D.C. wanted to set up an exhibition of antibiotics, including penicillin and then streptomycin, and they asked Waksman to send some artifacts of the original discovery, plus a page of his lab notebook describing streptomycin. And he sent down some lab equipment, rather primitive sort of shaking machine and a couple of toothpick things, and the exhibition was set up. And it said, here is the discovery of penicillin, and here is Alexander Fleming, and here is Selman Waksman, the discoverer of streptomycin. And then in 1973, Waksman died. And in 1989, a British microbiologist named Milton Wainwright got fascinated with this story. And he started looking into it, and he saw pretty much what I've just described to you. And so he wrote a piece in a scientific journal saying the moment of discovery actually belongs to Albert Schatz, and he was dumped on, so to speak. But this appeared to be a possibility for Albert Schatz to call the Smithsonian, gave him confidence, call the Smithsonian, excuse me, that exhibition that you have, not quite right. The Smithsonian, first of all, ignored him. Secondly, who is this person? Not on the roll of honor. Why is he sending us these notes? And then finally, they let him in. And Schatz said, actually, I can provide much better things than my professor. For example, the very first discovery that I've made of this actinomycet that produced streptomycin, I put it into a test tube and sealed it. And then I put the first amount ever of streptomycin in another test tube and I sealed it. And I took it to my mother on my weekend off. I took it to my mother, who never passed grade school, so she had no idea what this was about. And I said to my mother, at some point, all this is going to be very famous. Here are the two test tubes. So the Smithsonian, doing the right thing finally, said, great, we will display these test tubes. And do you have anything else? And he said, well, no, actually I don't. And he said, well, we have here some notebooks from Professor Waksman. And he said, well, how can that be? I mean, I know he used to check our work and he would copy into his own notebooks our work so that he would keep a record of it, nothing wrong with that. But at some point, some of these pages don't seem to add up. Like, actually, those two strains, if you remember, one of those strains that Waksman claimed was his, or that he had organized it, he said, that's absolutely not true. How did that appear? So anyway, the Smithsonian again did the right thing. So they took down things that might be anything wrong with them, and they finally reorganized the exhibition, and the case was closed as far as I was concerned at that point. Now, why no one had looked for the notebooks before, why they had been lost or not identified when the Waksman papers were archived, or not sent to the Smithsonian, or not included when the Waksman Museum at Rutgers was set up. What motivated Waksman to send a copy of his own notebook, not Schatz's, to the Smithsonian? All of these questions I leave with you to continue the debate, because it's not closed. There are still people who say Waksman was the discoverer, and there are others who say, no, nobody's saying Waksman is not part of the discovery. They're simply saying that he was not generous enough to include his student. All right, so I got 5 minutes. So just a couple of leaders into the question time. So how does this advance the cause of scientific investigation, and why should we be worrying about it? Well, just for background, the Nobel Committee has an almost impossible task. They have to choose a discovery that everyone thinks deserves the prize, and then according to the will of Alfred Nobel, they have to award it to, at the most, 3 people. In a world that today is so interconnected, and in sciences, especially like biology, where discoveries are often so complex and so dependent on the work of many more than 3 people, the Nobel Committee is almost bound to be criticized for leaving someone out. And it happens pretty regularly. Changes in the award are possible. The Nobel will is, they could increase the numbers, they could do something, but so far they haven't done it. So should we be concerned about what happened to Albert Schatz? I think we should. The Nobel Prize, after all, is the ultimate accolade in science. You may disagree with it, you may say people shouldn't have prizes, but it has a lot of good things attached to it. And it creates, for good or ill, a scientific elite, and it also is something in the popular mind. So it separates the receivers of the award from all other scientists, and it creates role models. There's nothing wrong with that. In the streptomycin discovery, there is one more point. The Nobel Committee did not feel it necessary to look at all the evidence particularly from the court case, that pointed to who deserved the credit in this case. One of the reasons that they didn't do that is because somehow the court case was, slightly beneath the idea of the scientific clergy. They only looked at scientific papers, and if it's not in the scientific paper, it doesn't exist. We could talk about that. And finally, in this case, the Nobel Committee revealed its own bias. And maybe this is changing, I hope it is, but I don't know, I don't talk to them on a regular basis, so. But there's a sort of class thing here which you can't really ignore. And it goes back to the Droit de Seigneur and the professor and who's on the paper first and all that. There's a Swedish word, I'm not going to pronounce it properly, medarbetare, which literally means with work. So it's the person who actually does the work. And when they, in the original Swedish, when they made the award, Schatz was a medarbetare. And he was not, as they pointed out, a collega, a colleague of equal standing to Waksman. So my friend Peter Lawrence of the Cambridge Medical Research Lab wrote that research students are like boosters on space rockets. They accelerate their supervisors into a higher career orbit, and then, when their fuel is spent, fall to the ground like burnt-out shells. Oh dear. Well, anyway, in the streptomycin case, Waksman never suggested that Schatz was worthy of more attention than his co-researchers, although the moment of discovery, I would say, belongs to Schatz. Thank you very much.
Thank you so much for a wonderful story with repercussions for now as well. We have a good amount of time for your questions and comments and some discussion. We have a couple of mics that we can bring to you. If you have a question or a comment, please identify yourself before you ask your question. So the floor is now open.
Oh, good. Any questions? Oh.
Hi, thank you for that. My name is Karen Knight and I'm in the medical library. Is there anything that you, in hindsight of course, which is always 20/20, would have suggested that Schatz do differently to not have had this happen in this particular flow? Any lessons to be learned for the future?
Terrific question. I suppose he could have gone to a medical ethics seminar to see what happened, see how these things occur, and therefore be prepared for it. He could have understood that both he and Waksman had chips on their shoulders about the other. Then there was a class thing, and recognize a class thing, you can usually get out of it or make it better if you know that it's operating. That. Otherwise, no, not really. He was a man obsessed, I think, with his research, and it's difficult to stop those people, and they shouldn't be stopped because we need them.
I may want to follow up on that just briefly. In reading the book, there were several excerpts that you included. Schatz remained in correspondence with Waksman for several years in the early part of his career after leaving Rutgers. And some of the excerpts that you include indicated to me anyway that Schatz was very much writing to a father figure. He was confessing a lot about the jobs that he had that weren't fulfilling in certain ways. Was he in some ways unknowingly playing into Waksman's hands in this in this?
I don't think so. I never really thought of it in that light. I have thought about it in the light that you mentioned first, which is, you know, Schatz was basically on his own. His father died young. He was looking after a new family. He was quite desperate to keep his job, to find a new job, and Waksman had been, you know, there was a time, undoubtedly, that everything was very jolly in Waksman's laboratory, and they had picnics down by the Jersey Shore, and Waksman made what he called kosher hot dogs with sausages wrapped in bacon, stuff like that. I mean, a man not without humour. And when that was playing out as it did in those early days, I think everybody had a nice time, and I think everybody who worked under him did look on him as the Great White Father, as they called him, and it was completely shocking, even more so because of that.
Hi, my name is Michael Dong. I'm a first-year medical student. You mentioned in the lecture that Waksman formed a foundation to help, spread his name, in your words, and kind of maybe helping him get that Nobel Prize. I was wondering what you thought about the impact it had on spreading streptomycin around the world. Did Waksman make a big change in getting that medicine to less privileged places?
Well, as you can imagine, when it was first discovered, there was a huge shortage. They simply couldn't keep pace with the demand, and particularly in England, I read a lot of correspondence of people writing saying, "Why can't we get the British Medical Association?" Blah blah blah. Those type of organisations looking at the possibility of getting more streptomycin from the United States, but the United States, it was all being run by the army, basically. And so it was very difficult. So in a sense, yes, those Waksman institutes were promotional for streptomycin, but they weren't producing anything. It was not increasing the supply. Does that answer your question?
Yes.
Okay.
Brad Worrell from the Department of Neurology. To what extent, taking what you presented as the facts of the matter, do you think Rutgers is culpable in this?
Okay. Yes, very good question, thank you. I hadn't talked about that. So, yes, is the answer. Why would they do that? Well, he was their only Nobel Prize for a long time, so it was obviously important for a small agricultural college to have this kudos. But also involved in the archive, we discovered this letter right at the end. I don't know whether you can read it, or would you like me to read it out and maybe not have a copy of it here, but basically it says that this is done by an administrator at Rutgers who is asked the very question that you asked. And he says, did we do something, could we have done something different? What are the motives behind this? And let's lay them out. And so this was the answer, that he wanted no reward for streptomycin, but the build-up went on. He got to love the public acclaim and the financial benefits, so that he forgot the Schatz name and acted badly, yes, when Schatz first asked in a letter and then demanded in his suit his share of the credit. So they could have, for a long time, they could have done something different. But eventually, after Waksman's death, Schatz was awarded the Rutgers Medal, which is the most honorable thing you can have. Somebody called me up the other day and said he's still not on the list of alumni. I don't know whether this is true or not. Maybe.
My name is Laura Alexander. I work in the college here at UVA and teach in religious studies. I had a question in part based on your description of the Nobel Committee as having an impossible task, especially in the sciences. And it does seem to me that questioning whether it's possible to give a Nobel Prize to between 1 to 3 people in a time when scientific research is shared, there's a whole community that's working on various projects together in some sense. The debate over Waksman and Schatz seems to be possibly part of a larger debate over whether we overvalue either the discoverer or the person who sort of led to the discovery or the moment of discovery, and undervalue kind of the community that's part of that, whether that's Rutgers, the lab director, the government, which is funding research because there's a wartime effort going on, and so on. So I wonder if you could just speak a little bit about how this fits into that kind of broad debate about the way that we value kind of the individual discoverer, the great individual.
Yeah, that's a great question, and we only have 2 minutes, okay. I think that, as I said, I think that, I'm not against prizes, right? Because it raises the standard, people look up to it, people work harder, but the way in which these are administered and the way in which they're chosen is forever going to be in dispute. So the question is, how can you have a list of criteria which will somehow ameliorate the dispute? And I think that the Nobel will is something which is very, very rigid, and you can't, you can't make it more than 3, and that's it. So something could possibly be done there. And the other thing is that if you are a PI, then in the modern context, why don't you talk to your graduate students, your postdocs, or whatever you had right at the beginning, and say, okay, if anything happens around here, if we strike lucky, this is how it's going to work. Everybody agree? Or shall we have the debate now rather than later? Something like that.
Maybe we have time for one more question?
Yes, excellent talk. Thank you for this. I'm curious what happened to Albert Schatz. I see you have a couple of his family members on your thank you list.
Yeah, he died in, can't quite remember exactly now, but his wife, Vivian Schatz, is still alive. She lives in Philadelphia. She was tremendous help in the research. He wrote up his version of the events, and before he died, and obviously they had to be matched with whatever was there. But he led a very simple life. He never really had a proper job after Rutgers. For 2 reasons, really. One was that, when he discovered what happened to him, it really shocked him, and he lost a lot of confidence. And he was not looked gladly upon by the scientific elite, if I may say so. And he also had some money. So the incentive for him to, 12,000 a year didn't sound very much now, but in those days quite a lot. So I'm not sure that he died a happy man, but he died after that encounter with Milton Wainwright. And so in a scientific paper, he had actually been, they'd agreed, you know, not a court of law, but a scientific paper, 1989, that yes, he was the discoverer.
Thank you.
Great, so thank you so much, Peter Pringle, for a very fascinating and sobering hour, and a cautionary tale for all of us. We hope you'll come back next week. It's a combination medical center hour, medical grand rounds. We have Dean Paul Worley from the medical school at Flinders University in Adelaide, Australia. The winner, speaking of prizes, the Brodie Medical Education Award here at UVA. And his talk will be on transforming clinical education, science, service, social accountability. So we'll see you then. Thank you all so much. And thanks to Peter.